I'm going to introduce our next speaker, Astrid Junker. So first I'll give you the facts. She has a diploma in nutrition, in nutrition from the Friedrich Schiller University from 2004, a PhD from Martin Luther University in Hull, 2009. She did a postdoc in bioinformatics and systems biology at IPK, finishing that in 2012, and is the scientific coordinator of the DPPN, which is the German Plant Phenotyping Network. This is also at IPK. She's a group leader in acclimation dynamics and phenotyping at IPK, and she's interested in how plants acclimate to climate change. The aside I will give you is if you find yourself at a meeting and you're somebody that is willing to stay up late at night, Astrid will join you.
Thanks, Carolyn. Thank you very much for this nice introduction. Thanks for the invitation. I'm looking forward to this evening. It's really nice to be here and to present the results, facilities, everything that's going on, not everything, but a part of it what's going on at IPK in Germany. And I would like to show you how we use our controlled environment phenotyping facilities for studying acclimation capacity in plants. And I would like to switch to the next slide. Okay. So I would like to start giving a very short overview of our institution, so IPK in Gatersleben. It's called Leibniz Institute for Plant Genetics and Crop Plant Research. And this is actually a very unique place because we have at IPK, on the one hand, 4 scientific departments which do a lot of interdisciplinary, basic, but also application-oriented research in very different fields, from plant morphology, cell biology, physiology, over genetics up to systems biology and bioinformatics. And on the other hand, at IPK, we are housing Europe's largest federal central ex situ gene bank, so housing several thousands of accessions of crop plants. So all these are maintained, characterized, and also given away to interested people from our site. And so the unique thing at IPK is that we basically can combine this research part with this nice infrastructure, or with these plant genetic resources. And this is basically one of our main aims, to efficiently exploit genetic diversity. And this is what we are doing on various levels, mainly on the genotypic level. So we have a large genotyping infrastructure, but, and this is what we want to talk about today, also doing phenotyping, so phenotypic analysis of plant genetic resources. And in terms of phenotyping at IPK, it's very important to mention again what Carol has said in the introduction, that we are a partner in the German Plant Phenotyping Network. So the DPPN, you might know these colors here and all the different PPNs. So you've heard about the NAPPN in the introduction today. So most of the phenotyping facilities were basically funded through our ministry in the course of this DPPN project. And so at IPK, we are doing phenotyping on various levels, starting at the cellular level with some microphenomics facility, where we really, on a cellular level, quantify plant pathogen interactions with an automated microscope, as you can see here. Then we do also phenotyping on the organ level, again looking at plant pathogen interactions, but on a leaf level, quantifying disease systems in an automated way on the leaf level. We have an NMR platform where we can, in high throughput, quantify seed composition as an example for an organ-based phenotyping tool. And then what I will mainly talk about today is phenotyping on the organism, so whole plant level, but also on the stand level. So we have platforms for whole plant phenotyping in controlled conditions mainly, but also a small platform, as you can see here, that works in the field. So all these phenotyping facilities are supported and accompanied by other infrastructures, or infrastructures such as a large image analysis pipeline. So we have a work group which is only dedicated to image analysis, and this just gives an overview of the tool that we are using for deriving features out of our whole plant phenotyping platforms. So it's very similar to what Bettina also has been shown before. We have several imaging modalities. We do a segmentation, and what is important to, as a take-home message from this slide, we look at, or we extract many different traits from our images, and this is not only kind of geometric architectural traits such as height, area, volume, convex hull, and so on, but importantly also a lot of color traits which are really important, especially when you're working in abiotic stress. So we derive pixel by pixel information on plant color, and also color traits from static fluorescence images or near-infrared images. The second infrastructure that is very important when you do phenotyping, whole plant phenotyping, is a data management infrastructure. And together with my colleagues Uwe and Matthias, who are shown here, and also Daniel, we are working a lot on phenomics data management. And this is the only slide I will show about that today, because I will rather focus on biology. But we can talk if you're interested about this later. So we have a LIMS system at IPK, and we basically have an almost automated workflow for experimental metadata collection, formatting in standardized metadata formats, combining it in a database with raw and result images, and then going through a data publication pipeline, having our own repository where everybody basically in the end is able to access raw data, result data, metadata, and standardized formats, and also conform with the minimum information standard. So MyIP standard has been introduced by Carolyn before. So this is actually a very important point, and you can find data sets that we have published already. So phenomics data sets with a standardized metadata format in the MyIP conform configuration. For example, in this paper here in Scientific Data, and the MyIP guidance has just been transformed basically to the next level. So there's new versions, and we are just about to publish new data sets with MyIP conform data, just for your information. So with this, I would like to get into a bit more technical details and describe shortly our whole plant phenotyping platforms. So these are the platforms that are up and running at that time. We have 3 conveyor belt-based systems similar to the one that Bettina has described in the phytochamber and in glass houses. So we have systems for small plants like Arabidopsis for up to 30 centimeters of height. We have a system mainly used for small grain cereals, barley and wheat, where we can phenotype plant up to 1.50 meters in height. And we have a large plant system where we phenotype maize up to 2 meters of height. These have different capacities. So in the small system, you can get from like 4,600 very small plants up to 1,600 plants here in the large system. We have different imaging modalities. We do RGB imaging, near-infrared imaging, static fluorescence imaging. We now upgraded these systems, at least 2 of them, the small and the large system, for root phenotyping, which works pretty nice, especially here, as you can see maybe from this image. Now it's hard to see. For large plants, we also upgraded these systems to do some physiological measurements. So we can also measure chlorophyll fluorescence using a pump type of measuring module. And importantly, we also do a lot of environmental monitoring. So we have upgraded all the glass houses with wireless sensor systems that can measure aerial conditions, but also soil conditions. So we can also measure soil temperature, soil humidity, and so on. We even now do the watering in this, at least in the large system, we do the watering based on in real time measured soil sensor values. So we also do envirotyping basically. And I think what's down here, this is all clear to you, why phenotyping, or what is the important features of phenotyping. We use imaging-based methods, thereby we are non-invasive, so we can quantify a lot of features which would otherwise not be accessible, and we can look at the feature dynamics. This is the important thing. Okay, so we do all that, obviously under controlled environments. If this was not the case, I would probably not be here today. But this is also very important because of the biological questions that we are addressing in my group, which is, as I said in the beginning, acclimation capacity. So probably it's, you are aware of that, but people always mix up as acclimation and adaptation. I just want to make it clear again here because it's very important to discriminate these two things. So first, what they have in common. So they have in common that they both enhance plant fitness and survival. So this is what they have in common. What distinguishes them is basically the time scale. So when you look at adaptation, then this is something that happens over generations. Yeah, so phenotypic adaptation is something that happens over generation when plants, for example, adapt to whatever drought areas in Australia. What we are looking at is acclimation. So this is processes that happen much faster. So we are really looking at short responses to changes in environment that lead to physiological, biochemical, or morphological modifications. So this is basically what we are interested in my group. And we are working on several different factors. So these are mainly factors that change, especially when you think of the field, that change rapidly. So temperature and also light intensity or spectral composition, but also water. So we are also looking at drought stress. We are looking at this in different plants, model and crop systems. We are working on small grain cereals. We are working on maize in terms of temperature and also light acclimation. And as I said, also in model plants, so we also worked on Arabidopsis quite some time. And for today, I would like to go through two use cases or two applications of our phenotyping systems. And the first thing I would like to show you is a very nice experiment where we looked at drought stress in Arabidopsis. And this is very important, this is a very important experiment from my point of view, because we here really tried to integrate phenotypic data with other omics data. So really looking at doing like big data science and trying to establish kind of statistical workflows or methods from a bioinformatics point of view to do data integration. That's why the experiment itself was actually very easy. So we only looked at one genotype. We just took the reference genotype Columbia 0. We put it in our automated system where we can nicely do drought stress regime because we have automated draining and watering. We had a high replication because over the time of the first phase where all plants were well watered, but also over the drought stress phase which you see here. So we really get down to 10%, this is field capacity, 10% field capacity. And over recovery, we sampled at all these time points for RNA-seq analysis. And this was done in collaboration with Andrea Bräutigam. And this is just some representative images over the course of the experiment. Top view Arabidopsis images of the well-watered plants in the upper panel here, and the drought stress plants in the lower panel. What you can see by visual inspection, you don't see much, only at the very end of the drought stress period. Plants look a bit smaller and seem to have a change also in color, but this is lost very fast during recovery. So you have to imagine we have these images like for every day of the course of the experiment, and we derive about 200 traits architectural traits, color traits, many different traits for every day of this experiment. And this is the statistical workflow that we came up. So Stefan is a bioinformatician in my group, and he did a really great job. So we started with some data transformation, and one of the first, or one of the most important steps of this workflow is this one here. So first, we wanted to see which of these 200 phenotypic traits would best classify a drought stress from a well-watered plant. So for this, we used a simple machine learning approach. It's called random forest. And when you do that, you basically come up with a list of traits. And this is not only traits over the whole time course, but traits per day of the experiment, and you get an importance value. And we have a long list, and we then basically group these traits into 3 groups. You see over here. So we have in red architectural traits, in green color traits from the static fluorescence imaging, and blue color traits from the visible imaging. And these bars here represent the cumulative weight factors, or importance factors for classification of these trait classes over time. Unfortunately, you don't see here anymore that the drought stress phase. So you have to believe me, the drought stress ends here, where we have now the pointer at day 30. So what was first nice to see that we get, as expected, the best classification at the latest day of drought, because at that time plants would be as different as possible, or most different from each other, drought stressed and well-watered plants. But what you also see is that the color traits, so in green and blue, they get important for classification much earlier than architectural traits. Because you see the color in the plant, you cannot see by eye, but the computer can see it. Color traits change much earlier than architectural traits, so a decrease in biomass you will see only later than a change in color. And also color traits stay more important basically after recovery. So you see here, after recovery, importance goes down rapidly. This is also expected, but for some color traits, they don't recover as fast as, for example, biomass, so architectural traits. So this was very nice to see. Then what we then did, so we came up basically with a list of 28 most stress-related traits, and we then looked at the time course of these traits and put these, integrated these with the RNA-seq time course data. And what we did was basically, we used again a machine learning approach to predict the time course of the phenotypic traits with the time course of the transcript traits. So which genes would basically be most predictive for the phenomics time course. And this was again done using a random forest regression method. And so basically in the end, we end up with a list of traits and a list of genes that are most predictive for this trait. And what we then did in order to visualize this, we basically came up with we just derived a network out of this with a trait and gene network. And this is just to show you how this will look like. So we have many traits, so 28 traits, which are important for the classification, and they will be connected with the genes that are most predictive of this trait. We then did an automated network layout to derive subnetworks. And this is what the network basically looks like. And we identified 3 different subnetworks, which was very nice for us in the beginning, which represent different trait classes. And we did not just lay out the network how we wanted it to be, but what was basically done through an automated approach. So we came up with 3 subnetworks, which were representative of different trait classes. So up here, we only have traits in red. When you remember from the classification before, red are the architectural traits. We have one subnetwork down here, which is represented by only color traits from the RGB imaging. And we have another subnetwork here, which is represented by the traits from the color traits from the chlorophyll fluorescence, from the static fluorescence imaging. So when you think of drought stress, you might know that there's different stages in drought response. So there's an early response, which is mainly, very early priming response, which is dominated by ABA accumulation and ABA signaling. Then you have an acclimation preparatory stage, and you will have a late acclimation stage. And we were really happy to see, when we look at the course of representative phenotypic traits in these three subnetworks, you see these three figures here. In red, you will always see the drought time course. In blue, the well-water time course. That these different trait classes and these subnetworks are representing different stages in stress response. So here, down here, you see this trait changes quite early during drought stress. It changes more drastically and also quite early. And then during recovery, it recovers quite slowly. You see here another color trait from the fluorescence imaging, changes also quite early, recovers very fast. Up here we have the architectural traits subnetwork. These architectural traits, they only change quite very late, and they recover also very fast. For example, biomass. So obviously, these three subnetworks of genes and predictive genes and traits were already representative of the different response stages, drought stress response stages. And what we did is we looked at geo-enrichment, so enrichment of gene ontology terms in these genes in each subnetwork. And this made us really very confident that this makes a lot of sense, that our experimental approach was very successful, because we could, for example, find down here in this subnetwork for color traits, we found enriched genes of anthocyanin production, flavonoid metabolic process, which makes a lot of sense because they are ROS scavengers. Anthocyanin would obviously change the color of a plant, so this makes a lot of sense. This is early response, so this happens quite early in stress. In this subnetwork for the plant fluorescence traits, we find, for example, chlorophyll biosynthetic process, but this is clear that this changes, so it's reduced also as an early response. Proline is known as a metabolic component which is increased a lot under drought stress. So this all makes a lot of sense, and even here for the late subnetwork, we found something like pyrimidine, metabolic processes, everything that is related to DNA synthesis, which also makes sense because plants stop growing, so they would reduce their DNA synthesis. So we were really happy with that, and we will now go on and not just look at things that are known. This is just for validation, but the most important thing now is to basically look at things that have not been known, that have not were not known before. We want to identify novel candidates and want to do functional validation of genes, predictive genes that we have identified through this process, and see if we can identify new genes or processes underlying drought response. Okay, so this was my first use case in Arabidopsis, and now I just shortly would like to step into what we are doing in terms of temperature acclimation in maize. And this is what we are basically doing in the large plant system. So here you see an image. You see maize plants are already quite big here. We can go up to 2 meters of height. We have the 3 imaging chambers here. And most importantly, we have upgraded this system for kinetic chlorophyll fluorescence imaging. So we have this adaptation area here where we can do light or also dark adaptation, and we have this FluorCam device, which is a large LED panel with a camera sensor in the middle, where we can do all the measuring light, actinic light, the light pulses, everything what you need for chlorophyll fluorescence. And we use this system. Henning was the one who published the protocols for use of this chlorophyll fluorescence imaging system. So we use this integrated system we asked the user to screen the maize collection that we have in our IPK gene bank, which has about 1,360 accessions. And over the years, we have selected around 80 interesting accessions, and we benchmarked these against a reference panel and also against commercial hybrids. And since we are interested in temperature response, so we have always done 2 experiments. One experiment under normal warm conditions, as it's up here, as Mails likes it, so all the time 25, 20 degrees. And we compared it to an experiment where we started out quite cold. We germinated them at 15, 10 degrees, then did 2 temperature shifts to end up with the same warm temperature here. And what we have seen when we look at all the different traits that we have quantified using our image analysis, we start here in the left panel. We see that the hybrids, which are the dark blue spots here in this PCA, they are kind of accumulating here at the right side. So you would expect that they would outperform the gene bank accessions. So they are quite different. But what was still interesting that there are some gene bank accessions which perform quite well compared to the hybrids. Yeah? So they're not too different under warm conditions. And the same, a bit more pronounced. I mean, it's hard to see, but here's also these dark blue symbols here, which again are the hybrids, which even outperform a bit more under cold to warm conditions. So this was nice to see. We have obviously some potential in these old gene bank accessions to look for. We then went on. And during these experiments, we always used the system for kinetic chlorophyll fluorescence imaging and screened these plants at different time points. And when you look at the warm experiment, and here, sorry for that, but there's another different color code. So gene bank accessions are in green, the hybrids are in red, and all reference lines in yellow. And this is growing degree days, so GDD. So over time, you see that for chlorophyll fluorescence, so operating efficiency, this is the trait that we measure, efficiency of the photosystem 2. There's not much variation, it doesn't change over time. There's a tendency here that hybrids perform a bit better, but it's not really striking. However, when we look at cold conditions, we see that. So there's a dramatic increase in variability in this trait at very early stages under cold conditions, which continues to be visible at this time point, but later on we see a similar picture as we saw here under warm conditions. So the question now is why are these hybrids, this is the second point, so you see that the hybrids outperform all the other accessions. So only the hybrids are here at the top end of the value distribution. So they probably obviously have the capability to have a much higher photosynthetic efficiency under cold conditions. So this was very interesting. And even then we went on to, again, do a classification using machine learning, classify between hybrids and gene bank accessions. And we even could see that among all traits that we measured, this early photosynthetic efficiency, which is this blue bar here, this is the trait which has the highest importance for classification between a hybrid and an inbred under cold conditions. And this was not having an importance under warm conditions. So this is really something that obviously is very important. And it's much more important because we could also see that it's highly correlated with a late biomass. Yeah. So the early operating efficiency, I know probably it's hard to see, correlates quite well with the late biomass. So it also has some relevance, ergonomical relevance. This may be hard to see, but this is a correlation matrix, and these columns 1 to 4 are the operating efficiency values, and all the other columns are the biomass, plant biomass that we derived from our imaging over time. And what you can see is that the early operating efficiency correlates quite well, and the correlation even increases with the time. So it does not correlate quite well with the early biomass, but it correlates much better with the late biomass. So it's even more, this made us even more confident that this is something to look into. And yeah, this is what my new postdoc will now start to analyze in more detail, which influence has low temperature on the inquiry of photosynthetic competence, because we think that this is something that has to is related to early photosynthetic competence, and maybe also with the timing of the switch from hetero to autotrophy. So this is something that we will analyze in more detail. And what we also are looking into in maize is how maize is acclimating to light switches, so to fluctuating light. And for that, we again used our chlorophyll fluorescence imaging system, and we employed a more complicated protocol, and we looked how fast could plants activate their photosynthetic machinery from darkness at the onset of light, basically. Yeah. So we measured them in darkness and then turned on the light and looked how fast would they come up to activate their photosynthetic efficiency. And when we did that over the panel of hybrids and inverts that we had analyzed before, I should not leave out this. So when you do chlorophyll fluorescence imaging, there's 3 ways where your electrons can go to. You have the light energy. They can either do photosynthesis, or they can yield in non-regulated loss processes, so basically fluorescence or heat. And all this sums up to 1. Yeah, so that's why we always add up to 1 here. But this is the yields of this heat and fluorescence dissipation and chlorophyll photosynthetic processes down here in blue. So you see that there's quite some variation in how fast lines can activate photosynthesis in blue, but obviously the hybrids, which are marked down here with these black arrows, they are not better when you look at this trait. So there's some potential, obviously, for improvement here. And this applies to a switch from dark to light. What is even more important when you think of a field where plants are always subjected to light switches, to look at switches from low light to high light. And this is what we have done also with all our accessions. Yeah, so we basically look at operating efficiency at low light, then we switch on high light, and then get back to low light again. And you see at using the example of these 2 reference genotypes, B73 and MO17, that there's quite some variation in how fast they can get up their photosynthetic machinery. And this is actually very interesting. And we have also seen a lot of variation here in our gene bank accessions. And this is actually something that we want to analyze in more detail. And this is what we need facilities for, where we can do fluctuating light under very field-similar conditions and in a very controlled and reproducible manner. And that's why IPK has invested into a new plant growth facility. And this is the idea which has been initiated by Thomas many years ago. So we want to do actually phenotyping in defined or designed relevant, field-relevant environments. And this is our new building here. So what we want to do there is we want to simulate field-like conditions. Yeah, we want to do controlled fluctuations in order to be able to reproduce field-similar conditions also. And we want to elicit a trait expression that resembles that of typical field situations. We want to run specifically designed cultivation scenarios which are highly reproducible. We don't want to simulate exactly what's happening outside in the field, but we call it field-relevant conditions. We want to do phenotyping, this is clear. We want to dissect complex traits and their dynamics. And we want to also simulate future climate scenarios. And that's why this building has a quite sophisticated climatization infrastructure, which allows us to go to have an illumination which is very sunlight similar, and can get up to 1,500 micromole on the plant level. So we have quite a wide range of air temperature, humidity, and CO2 concentration that we can run. We can also simulate wind, and we can regulate all that with fast changes in order to really be able to have a field similar setup in there. This is how it looks like. So we will have these 4 compartments, and if it works, I hope it works, then we can look at it in this.
The plant cultivation hall will comprise 2 different phenotyping systems in an area of almost 500 square meters. One is a container-based system for plant stands, the other a so-called rhizotron system for individual plants. The rhizotron system is being developed and implemented in collaboration with the Forschungszentrum Jülich. It will support the simultaneous cultivation of up to 360 plants and will enable very detailed studies of the root architecture. Imaging towers equipped with specialized lighting and camera systems automatically move along the rows of rhizotrons. They successively take out individual rhizotron boxes to image the plants. The images are digitally evaluated, providing information on the characteristics of the root architecture, as well as on root and shoot growth.
One half of the building, what's more field similar, is this.
The other area of the hall houses the container system for the phenotyping of stands of plants of vegetation. Various camera and sensor systems are mounted on a robotic crane that can be precisely positioned. These plant imaging systems use different wavelengths and scan them in 3 dimensions. The soil in the containers resembles the natural soil structure in the field. Its temperature can be precisely controlled at different depths. The temperature and soil moisture are monitored by numerous sensors. Built-in imaging tubes allow the recording of root growth processes.
Sensors for root phenotyping and also soil sensors in order to measure soil conditions. And this is, so the building is now ready, and we have done the first proof of concept experiment. And for that, we have chosen 11 maize genotypes that we have grown in the field over 3 years. In the same setup as we have done it now in these containers. You see these double rows with each 9, so 9 plus 9, 18 plants per container. We have phenotyped them manually throughout these 3 years, and now also in these containers in the plant cultivation hall. So we measured plant height, leaf angles, V stage, developmental stages, all different parameters. Has been done by Mark and Thomas Group. But the most important thing here is that we have measured weather data throughout the 3 years. So we know exactly at which day, basically, the sun was shining, how was the rain regime, how was the humidity of the air. And from these weather data of the 3 years, we defined a setup for the climatization in this proof of concept experiment. So we basically used for every week in the field the median value as a kind of normal day, and from that we derived a good and a bad day with the fluctuations throughout the day, and also then the seasonal variation. And this is how it in the end looked like in our plant cultivation hall. So we have a lot of fluctuation, as I said, throughout the day, but also seasonal variation. And I'm not able to show the data, or we are still in processing of the data, but you have to believe me, and you see some pictures down here. So we grew these maize plants up to maturity, and they really resembled much more the field grown plants than the control plants that we had in parallel in the glass house. So they were kind of much shorter, had thicker stems, not like the ones that we have in the glass house, and also from the plant height progression, they looked very much, resembled very much the field grown plants. So we were really happy that the concept basically was proven with this experiment, but it needs a lot of optimization. So this is the very first experiment. We now have to build in also all the phenotyping systems also. So there's a lot of work going on there. And with this, this final slide, so we are involved in all the different PPNs from the German level down here over the European levels, where we now have the EPPN 2020 project, for example, where people can access our facilities, at least the ones that are in operational mode already. And we are also members in IPPN and coordinating this controlled environment plant phenotyping group. And yeah, with this I have to thank a lot of people, as the previous speakers have said. It's a combined, a consortial effort somehow. So I have to thank especially Thomas, because he was the one starting with all this phenotyping at IPK, Mark, Henning, Stefan in my group, our image analysis guys from the gene bank, guys from the IT. We have a lot of engineers here and mechatronics guys. So yeah, and in the end, I have to thank also my kids, who are always looking forward when I'm back. And I would like to thank you for your attention.
So I was interested in, I understood the original separation of the network into 3 subnetworks. And then, but when you were talking about the GO enrichment analysis, the little inset graphs there. Mm-hmm. So I was puzzled by the difference in the patterns you see there with the overview graph of the,
okay.
What is being represented in those small inserts?
I didn't explain that, yeah, sorry. So basically this is, so the RNA-seq data was, underwent hierarchical clustering, k-means clustering.
Okay.
And we identified about 16 different clusters.
Okay.
And we did an over-enrichment analysis also for genes in these clusters. So most of these genes in this subcluster were derived from this k-means cluster of the transcriptome analysis.
Okay, that's an expression
so
plot.
this is expression data,
Okay.
and this is, I mean, it should be quite similar to the other part. I think there's just so the temporal scale is a bit different, because here this is basically cut shortly after this one here. Yeah. So because this is so these transcript profiles were used to predict the phenotype, so they have to be similar.
Okay, yeah.
So this is.
I missed that. Okay, thank you.
So those are great relationships, you know, and it shows that, you know, you can demonstrate correlations, you know, between these various parameters and let's say biomass. I guess they aren't causalities, though. In other words, because you're having stress conditions, you know, you're losing things are becoming chlorotic, etc. But the reason they're becoming chlorotic may not have anything to do with their ultimate effect on biomass. It's just that they're all being subjected to the same sort of stress conditions. And so you're losing chlorophyll, you're losing biomass. They show a correlation, but there's not necessarily causation, you know, between the, it may not be that because you're losing chlorophyll is the reason why you're losing biomass.
No, this is true. This is true. I mean, we don't. We think it's causality in terms of the gene-to-phen relationships in each sub-network, because this is kind of. I mean, we see, for example, the color traits down here. This is something that changes early. So we find GO terms enriched, which really nicely fit to the trait that we see. So I think these genes probably would really be important for color pigmentation of the plant, which we see under drought stress. But if this, I mean, I don't claim that this is the cause for a reduction in biomass that we see later on. So this is has nothing to do. So I think this would be too far.
Well, the correlation and the predictability that comes from this is really quite valuable.
Yeah, that's exactly. This was our main message and we used this kind of simple, well-described use case to drought stress and Arabidopsis to start with that because it's so well described that you can validate, as we have seen, that you can validate nicely. And we just wanted to really get into integrative data analysis because, I mean, I'm not aware of somebody who has done something like that, like predicting phenotypic traits from the transcriptome. And we wanted to do this for a long time, but we started with this use case just because it's so easy and it's well described. And we think and we are confident that there is much more than this. I just showed you what is described and how it could be validated with the known things. But I'm confident that there's much more in it which has not been described so far.
It's very impressive.
Thank you.
Thank you for this presentation. I just wanted to ask you, did you think, or you mentioned something about staging the stress? So you have like, can you think of biomarkers? Use the transcriptome as a biomarker for stress staging?
Yeah.
Have you seen those type of correlations?
Yeah, this is what you see actually here. I mean, we have this is, for example, a trait which changes early, so it would be an early stress marker from the phenotypic level, and the genes that we have here would be transcript biomarkers basically for early stress.
Right.
And the same up here for later stress phases.
And did it make sense if you look like on which biochemical pathway it maps? Does it make sense from perspective of plant metabolism, like certain pathway goes up, certain pathway goes down? Like for example, have you seen increase in proteolysis or decrease in translation?
There's a lot more GO terms enriched, and there's a lot about sugar pathways, which all make sense. I just didn't put it because it would be too complex. But there's a lot of changes in metabolism which we can see from this approach and which also perfectly make sense. Protein modifications also, yeah.
Let's take one more question. I can look also, we may have some questions here. Nope, no questions from the Zoom. One in the back, Trevor.
All right, phenomenal topic. Really amazing things that you're doing here. So I was wondering about the recovery. Is that part of the transcriptome and phenotype part? And if so, what are your thoughts about the temporal aspects of that? Because intuitively, you would think that the drought stress is happening much more slowly over time, whereas when you're giving the water back, it would be a faster response.
So it was covered from the phenotyping point of view. So we phenotyped all these. We also took samples, but we just subjected to RNA-seq now the samples until the first day after recovery. So we don't have the recovery dynamics covered in the analysis so far. But we have all the samples and we have all the data, so we probably would do that. We'll do that.
Okay, with that, let's thank Astrid again.